Welcome everyone to the DeFord Lecture Series. My name is Elizabeth Catlos and I'm a faculty member in the Department of Geological Sciences, which is part of the Jackson School of Geosciences at the University of Texas at Austin. The DeFord, or Technical Sessions Lecture Series, has been a requirement and a tradition for all of our graduate students since the late 1940s. It is our forum for lectures from distinguished visitors and members of our community. The lecture series is named after Professor Roland DeFord, who joined the university as a professor in 1948 with the purpose of enhancing the quality of the graduate program of the department. Our speaker will be introduced by Dr. Rowan Martindale, who is an associate professor in our department.
I'm thrilled to introduce Dr. Melissa Kemp, who comes to us from so far away as the biology department. Melissa is actually a joint appointment, so she's in the integrated biology department, but she's got a zero-time appointment here in the Department of Geological Sciences. She got her PhD in biological sciences from Stanford University in 2015 and her BA in 2010 from Williams College, which is where a friend of mine, Phoebe Cohen, is currently at. And her research is all about how environmental perturbations drive extinction, diversification, and colonization in vertebrate communities through time and space. She focuses most on more recent time periods and how sea level changes influence Caribbean communities. And so I'm excited to hear about her research. Take it away, Melissa.
Great, thank you for that introduction, Rowan. And thank you all for coming to the talk. I know many in DGS because of my appointment there, but I look forward to meeting more colleagues over the years. So I'm going to be talking about biodiversity in the Anthropocene and a paleobiological perspective on biodiversity. And since I'm talking to an audience that is more geologically inclined, I do think it's important that I first talk about what biodiversity is and why biodiversity is important. So biodiversity is the variation in life on the planet. We often think about it as the organisms that are found in an ecosystem at a given time point. Biodiversity loss is one of the biggest challenges facing humanity today. And this is because the loss of species threatens various ecosystem services that we depend upon, like food production, pest control, pharmaceutical development, and also nutrient cycling, to name a few things. And also biodiversity nourishes varied cultural, spiritual, and recreational practices that promote physical, mental, and emotional well-being in our societies. So there's an urgent need to characterize existing biodiversity, quantify recent biodiversity loss, and to determine how species and ecosystems will be impacted by ongoing and future global change. And in terms of my research interests, I study how vertebrates respond to global change phenomena like the ones in this figure. I'm mostly interested in detecting responses to climate change, invasive species, or as I like to refer to them, introduced species, and also habitat loss. And I'm interested in studying the impact of these phenomena on processes like extinction, colonization, and diversification, both spatially and temporally. I work primarily with lizards in the Caribbean, and the majority of the work that I'm going to talk about today is from that system, although we are starting some really exciting projects in Texas that I'm going to offer a little glimpse into as well. But insular systems like the Caribbean are extremely fascinating because islands are often biodiversity hotspots. And biodiversity hotspots, for those of you who might not be familiar with the term, are regions that make up a pretty small proportion of the Earth's surface collectively, but contain a large amount of species. And many of the species that you find in biodiversity hotspots are endemic, meaning that they are only found in that particular region and nowhere else in the world. And that's certainly the case for many of the lizards that we find in the Caribbean. And the ecology and evolution of many of the extant Caribbean lizards has been very well studied by scientists, especially scientists from North America, just because of the proximity of it to those of us in this continent. But there's much less known about ancient lizard communities, even though there is historic and paleontological evidence that extinction has taken place, as well as colonization taking place alongside these extinction events. So today I'm going to talk to you about my work and how the lack of knowledge we have about extinctions and ancient lizard communities more broadly impacts our understanding of how these communities are structured today. And this therefore has implications for the ongoing and future conservation of these biodiversity hotspots. So two things I hope you all get out of this talk is that paleobiological data provide long-term perspective on urgent contemporary issues, and that mitigating the negative impacts of global change requires interdisciplinary skill sets. And the thing about paleobiology is that it is innately interdisciplinary. It bridges biology with the geosciences, and it's very common for paleobiologists to pivot between these disciplines. So in my case, all of my degrees are in biology, but I have a lot of formal and informal training in the geosciences. And members of my research group come from both disciplines and often have a bachelor's degree maybe in the geosciences, but then are pursuing a PhD in the biological sciences. But it's broader than that. So paleobiology also has a lot of connections to other fields, such as archaeology, and certainly on the timescale that I work on, that's very relevant. It's also connected to history, law, and I do make note often of distinguishing genomics from other areas of biology, because there's been pretty recent interdisciplinarity between the paleobiology, genomics, and archaeology that have led to numerous innovations in methods and knowledge transmission across these fields, particularly our ability to get genetic data from fossils. And I regularly work with scholars in these areas as well as other areas in approaching these questions about global change and how past global change can inform future instances of global change. So now I want to spend some time talking about the Caribbean fossil record, just so that you have a little bit more familiarity with this system. The Caribbean is a geologically complex region composed of the Greater Antilles, which includes Cuba, Hispaniola, Puerto Rico, and Jamaica. We also have the volcanic island arc of the Lesser Antilles east of Puerto Rico, and then we have the Bahamas north of Cuba and Hispaniola. Because of the heat and humidity in this region, fossilization is pretty poor across the region. We lack a deeper time fossil record here. For lizards, my study taxa, we have a few fossils that are known from Miocene aged amber, like this specimen here. And those fossils are relegated to the island of Hispaniola, which is where Haiti and the Dominican Republic are. These specimens, while rare, have played an extremely important role in understanding diversification rates in biogeography, particularly for this one radiation of lizards, the anolis lizards, which has over 150 species in the Caribbean. And I can go on and on about those lizards, but I won't be talking about them too much today. But anyway, these anoles specimens in amber and other organisms that have been found in amber have played an integral role in understanding biogeographic processes in the Caribbean. But because of their spatial limitations, they have much less utility compared to the quaternary age fossils of the Caribbean, which is what I specialize in studying. So while this only encompasses the most recent sliver of geologic time, the quaternary fossil record is much more widespread with greater spatial, temporal, and taxonomic coverage. So every island in the Greater Antilles has quaternary deposits, although they've been quarried at differential rates. Many islands in the Lesser Antilles do, particularly the northern Lesser Antilles, which has a lot more uplifted reef and limestone than the southern part of that arc. And we also have a lot of deposits in the Bahamas. Most of these deposits are found in caves, and here's an example of an excavation that we conducted on Guadeloupe Island in the Lesser Antilles. These caves have very clear stratigraphy, and we're able to reconstruct funnel assemblages through time in these localities. Another great thing about the quaternary fossil record is that we generally know when some of the extinction and colonization drivers were operating and shaping these communities were occurring. So, for example, there was significant climate change during the Pleistocene-Holocene transition 12,000 years ago, and sea level rose to today's current levels. So in this figure, you have this light mint color that shows approximate island area and bank sizes at the last glacial maximum. And then in the green color, we have those land mass areas today. And what this means is that island area was greater in the past, and land masses like Florida here were much closer to one another. So here, Florida and the Bahamas were really close in the past. This means a few things. First, species richness may have been higher in the past due to what we call species area relationships, whereby larger land masses can harbor more species. And an analogy is thinking about a room. Certainly in pre-COVID times, the larger the room, the more people you could have there. But then we can also think about colonization rates. colonization rates may have been higher in the past due to increased connectivity of terrestrial habitats. So all of these small banks that we have presently in the Bahamas, or these small islands that we have presently in the Bahamas, were connected previously. These sites also document the arrival of humans into the Caribbean in the Holocene, the ways in which humans modified natural habitats and the species that they introduced during this time interval. And all of this is critically important because it's the only time in geologic history where the main drivers of biotic change are both humans and climate. Thus, the Pleistocene and Holocene provide insight into what we might expect during our current epoch, the Anthropocene. The Anthropocene is marked by humans being the dominant influence on climate change in the environment. We expect to see increased habitat modification, increased species introductions, global warming that will continue to change sea level, and more intense storm activity, some of which we're seeing today already. All of these factors will impact today's biodiversity, and the fossil record can give us insight into how these factors have impacted biodiversity in the past. What's especially exciting about the Quaternary records compared to more deeper time fossil records is that many of the taxa that we're able to describe from Quaternary deposits are extant ones. So we can reconstruct perturbation response trajectories for species and individual populations using data from before, during, and after the environment changes. And we also have contemporaneous data from extinct taxa and extant taxa, so we can compare and contrast how different species respond to the same threats, which may be instructive for understanding future responses and what species may persist when encountering environmental perturbations and which ones may perish. So for the rest of my talk, I'm going to present vignettes of my work on how biodiversity is shaped, not only by extinction and colonization, but also by the persistence and diversification of native species in changing environments. And while I'll be highlighting each of these processes individually, it's extremely important to remember that these processes are almost always happening simultaneously. So first, I'm going to discuss extinction and some of the early work I did on this front. And in my research, I've focused primarily on the relationship between extinction and body size for a few reasons. So compared to some other traits, body size is recorded in the fossil record pretty well, or it's easily estimated from fossil materials. But then also from a life history perspective, body size plays an extremely important role in life history traits. So, where a species can live, what reproductive output will be, what metabolism will be, and things like that. There have been a number of studies in a variety of taxonomic groups, both marine and terrestrial, indicating that extinction is size biased. And generally, there's a tendency for large bodied species to go extinct if you look at those data sets. But prior to my study, very little was known about the relationship between body size and extinction in lizards. There certainly was interest in the topic, and previous studies relied on metrics like the IUCN Red List to get a sense of extinction. But those methods have biases innate in them. First and foremost, not all species have been evaluated by the IUCN, and there may and certainly is an overrepresentation of larger charismatic lizards in that particular database. But if we were to use a fossil record where a true dichotomy exists between what is extinct and what is extant, we might be able to start articulating the actual extinction trends that exist. So I set about to do that in one of the systems that we have a pretty well-described fossil record for. So I created a database for all Caribbean lizards past and present. This database includes snout vent length, which is a commonly used body size metric, and I took these data from the literature. Body size estimates of extinct species are sometimes reported in the literature, but these fossils are often disarticulated. So we're not finding lizards preserved like this. We're finding individual skeletal elements like a dentary here. So with that, we are estimating body size, and the estimates that we generate are derived from body size estimation equations that we make using comparative material of extant related taxa. And to do this, what we do is we'll measure certain osteological characters that we have present in our fossils, like the length of the tooth row in a dentary. And then we will use that measurement in the known body sizes of the specimens we're measuring to create an equation that will allow us to estimate body size for fossils where we have that character preserved. So after his database was created, I noted whether a species was extinct or extant. I also noted the family of each species because life history is important, and the shared life history traits of closely related species might predispose some lineages to extinction. We had about 416 lizards in this database, about a little less than 5% of all lizard species that have been described worldwide. And in this database, 13 of those species are extinct. And once I did that, I looked at the distribution of extinct species shown in red and found that it was significantly larger in body size than the extant species, which are shown in white. However, we did find that extinction is not evenly distributed across the phylogeny. So there's a significantly higher proportion of extinctions in one family of lizards, the Lyocephalidae, than we expected. These lizards, in fact, are completely extirpated from the Lesser Antilles, and I'll get back to that a little bit later. It's important. And here is a museum specimen of one of those extinct species from the Lesser Antilles. Within Lyocephalidae, though, if we look within that family, extinct lizards are significantly larger than the extant ones. And there are 4 other families that have confirmed extinctions, and in all but 3 cases, the extinct species are among the largest, though the difference is not significant there. The one exception to that is the iguanidae, the iguanas. It's worth noting that these are some of the largest lizards in the Caribbean. All except for the introduced green iguana is endangered. And the largest species in the Caribbean is highly extirpated throughout its historical range. It's also important to mention that many of the species in these families here have been understudied. There aren't data for a lot of them in the IUCN Red List. So it's really important that we collect additional data on extant populations of species in these families so that we can continue to address what we see to be the non-random nature of extinctions in this archipelago that we're starting to uncover by looking at the fossil record. So this work really represents a first pass at trying to delve into extinction patterns. There are a lot of interesting directions that we are able to go in from here. And I'm going to outline some of the stuff that we can do or are interested in doing. One thing that we would benefit from is more detailed extinction chronologies, which help us narrow in on exactly when a taxon went extinct and what may have caused the extinction. So this has been a major focus of research in non-volant Caribbean mammals, many of which you can see here. Many of these you would not see on the landscape today. They have undergone significant rates of extinction. But for the Caribbean mammals, which have been a system studied quite widely, researchers have been meticulously radiocarbon dating fossil specimens to produce last occurrent date estimates. And when combined with body size data and archaeological data, these last occurrence dates show some interesting trends. So first, we see that there are 2 distinct waves of extinction. The first wave of extinction coincides with the arrival of indigenous groups to the Caribbean, and large body taxa like monkeys and sloths disappear. Then we have a second wave of extinction that occurs when Europeans colonize the Caribbean. And during this time, we start losing small body taxa like the endemic rice rats and Nesophantes, which is a shrew-like insectivore that looks a bit like this Nesophantes, or this Seliodon here. Those are lost. Unfortunately, we don't have this type of temporal resolution for the Caribbean lizards yet. Many fossils haven't been radiocarbon dated, but such data sets would really help us disentangle the role and timing of these extinctions and how climate change and humans colonization events may be leading to extinction events that we're seeing. But with that said, I would like to point out that we have museum specimens for a number of the extinct species that were present in the Caribbean. So this really drives home the fact that at least some of these extinctions have happened on our watch when people were there to collect specimens. We can also think about other life history traits that might predispose lineages to extinction in the Holocene, in the Anthropocene. Some of these traits include habitat affinity, and there have been some researchers who have already started to look at these traits in combination with natural history records. And we see interesting patterns emerging there as well. So we can take, for example, the extinction of various skink species in the Caribbean, and here's an example of a skink. Those extinctions seem to coincide really well with the introduction of the mongoose, which was introduced during the colonial period to control rat populations in sugarcane fields. And it didn't really do much to stave off the rats, but instead they preyed upon and wreaked havoc on native reptile, amphibian, and bird populations, and particularly these ground-dwelling species like the skinks. We can also start to investigate the ramifications of these extinctions on other aspects of the ecosystem, which is something that we've started to do with regards to looking at community structure. And I'm going to go through an example of this work that we've conducted in an early Holocene assemblage from the island of Anguilla. So Anguilla, shown here, is a small island in the North or Lesser Antilles, and it harbors this early Holocene site at Cartouche Bay. During the late Pleistocene, when sea level was lower, Anguilla was connected to St. Martin's and St. Bart's as part of a much larger island. And with such a dramatic loss in island area, we might expect extinction from island area loss alone because of the species area relationships that I alluded to earlier. So this site had been described and excavated previously, and I revisited the materials. And in revisiting the materials, I found that the early Holocene lizard community was much more diverse than a previous study had indicated. So previously, only 2 lizard genera were described at the site, Anolis lizards and Lyocephalus, the curly tails I talked about earlier. But we also identified fossils from 2 other genera, Phalidiscalcis, teated lizard, and Thecodactylus, which is a gecko. But despite this diversity, the ancient assemblage is only a subset of present-day diversity on Anguilla. So there are 7 other species that we find in the island today that we do not have evidence of in the Cartouche Bay fossil record. Some of these taxa are introduced. I'll note that Iguana delicatissima, there has been some debate as to whether that one was introduced or not, but it's worth noting that it's only known from archaeological deposits, and it's also known to have been a food source for indigenous people, as if you look at its name, delicatissima, you might infer that. We also have some extremely small lizards at this on this island, like the Spherodactylus geckos. These are some of the smallest lizards in the world, and it's possible that during the screening of sediments and materials from this site, those bones were missed. But I also will mention that geckos are notorious for having a poor fossil record globally. I'll also note that the paleo environment of Cartouche Bay is relatively unique for the island. It's much more humid and densely forested than the rest of the island, which might also explain why we're not finding some species there too. But back to the 4 genera that I did find there. So 3 of those genera, Thecodactylus, Phalidiscalcis, and Anolis, are still found on Anguilla today. But Lyocephalus is extinct on the island. So this is a great system with which we can assess how the removal of a species might affect the rest of the community. So I calculated the evenness of the Cartouche Bay community through time using a metric called Pylos evenness index. Evenness refers to how close in proportion the number of species in a community are. An even community with 4 species in equal proportion will look like this, and will have an evenness index of 1. When evenness decreases, 1 or a few species begin to dominate, and the metric also decreases. So here is the evenness of the Cartouche Bay assemblage through time. I've plotted the last occurrence of Lyocephalus here in red, and that occurs about 6,600 years before present, which I will note is prior to human colonization of Anguilla. One of the first things that we see is a 53% drop in evenness between 9,000 and 8,000 years before present. And when we see a drastic decline in evenness like this one, it usually signifies that there's a loss in stability of the community. And indeed, it foreshadows the last occurrence of Lyocephalus at Cartouche Bay that occurs soon after. So after this last occurrence, Anolis becomes the most dominant species, consistently accounting for 80 to 90% of the population, while Thecodactylus and Phalidiscalcis persist, but a much lower abundance. So when I saw this dominance that the Anoles exhibit in the absence of Lyocephalus, I started to wonder, and I was really informed by ecological literature actually, was the loss of Lyocephalus a predator release for Anolis? So we know that Lyocephalus eats anoles in places where the 2 genera co-occur today, and here is photographic evidence of that in Cuba. But there's also empirical data from ecological studies of Lyocephalus and anoles in the Bahamas, which confirm that Lyocephalus has a negative impact on anoles. So Lyocephalus has been introduced to a number of islands with anoles, and those islands are represented in this graph with white circles. And in the presence of Lyocephalus, anole density decreases relative to islands without Lyocephalus. Also, when Lyocephalus is present, anoles are known to perch higher in the vegetation, presumably to help avoid interactions with this ground-dwelling predator. So an implication of this paleontological data, together with the ecological data, is that the high densities anoles reach today in some areas, especially in the Lesser Antilles, where Lyocephalus is completely extirpated, may be the result of the extinction of predators in this system. So the last comment I want to make about our extinction data is whether or not our results are specific to the Caribbean or exemplify a global pattern. And excitingly, there has been subsequent attempts to characterize extinction in reptiles more broadly using reptile fossil assemblages from around the globe. And these researchers, when doing that, found that extinct lizards are large-bodied and concordant with my data from the Caribbean. They found no trend for snakes. Extinct crocodiles are small-bodied, interestingly, and extinct turtles are large-bodied. And also because these researchers amassed a global data set, they were able to do something that I didn't do. They were able to compare extinction patterns in islands to those on continents. And in doing so, they found that there's a higher incidence of extinction on islands than on continents. And if we take a step back and think about this from an island biogeography perspective, this finding is not surprising. So prior to human colonization, many islands were predator-free. Extinctions on islands worldwide have been linked to human colonization and the accompanying habitat destruction and species introductions that occurred when humans arrived. So it's not very surprising to see that. But while there may be higher extinction rates on islands, in my opinion, it's also very likely that we have simply undersampled ancient reptile diversity on continents, and therefore we've undersampled extinction on most continents. And I'll tell you why right now. So here's a map of reptile species richness, and if you look at it, these red areas is where it peaks. It peaks in places like the Amazon, have a lot of it in sub-Saharan Africa too. In Australia, it's pretty high. Actually, in the southern part of North America, it's pretty high too. But we have a lot of species richness in these continental systems. But it turns out that many of these continental systems, particularly places like Australia and North America, researchers historically have not looked at reptile fossils as critically as they have looked at other taxonomic groups. So in studying those other taxonomic groups, most notably mammals, we see that there were significant extinction events that occurred during the Quaternary. So it is possible that reptiles like lizards persisted through these time intervals in these places relatively unscathed. But before we can say that, or say that extinction rates are higher on islands, we really need to look at these continental systems more critically and really evaluate what species are present. So our lab is starting to address some of this, and I'll get to that a little bit more at the end of my talk. But I think the takeaway here is that this is a really good example of why it's great to think about paleobiological data and try to contextualize it more broadly, but we also have to be really careful when we're doing that. So if we're incorporating paleobiological data into large-scale diversity studies, we also have to consider the context in which those data are collected and the historical biases and what is and is not studied. And that certainly is the case. If we look at the global distribution of extinct reptiles, it does reflect historical patterns of study a bit. So now I want to switch gears and talk about our work on species colonization. And I'm going to specifically refer to some of the work we've done in a paper that came out a little around this time last year in the Proceedings of the Royal Society B. This work is a collaboration with a former undergraduate of my lab, Jenna Wadman, and then Alexis Mikhailu of Middlebury College and Amy Goldberg from Duke University. And this is a study of the paleontological and archaeological evidence of human-mediated species introductions. And we used literature from both of those disciplines to create a database of species introductions for the Caribbean, focusing on terrestrial vertebrates at this time. And we delved into the ramifications of these introductions on present-day Caribbean biodiversity. This particular figure highlights only the taxa for which we have a first introduction date. And unsurprisingly, we see that the number of introductions increases towards the present in every single taxonomic group that we have data for. We define 3 time periods of interest for the study. The indigenous period, which is prior to European colonization. We have a colonial period and then a modern period. And we delineate the modern and the colonial periods from one another with the Industrial Revolution and the significant economic and demographic changes that were occurring in the Caribbean around the time of the Industrial Revolution. So we can look at these data more deeply and ask about the geographic origins of these taxa and why they're being introduced. And when we do that, there are a number of patterns that begin to emerge. So in this figure, each line represents a species that was introduced to the Caribbean, colored based on when it was introduced. It scales down here. The top represents the biogeographic realm from which it originated, so the Neotropics, Nearctic, Afrotropics, Palearctic, so on and so forth. And the bottom represents where in the Caribbean the species was introduced to, so the Bahamas, Greater Antilles, or Lesser Antilles. Here I've only applied the taxa for which we do not have an introduction date. As you can see, this is a pretty significant number, although it does vary by group. We can then overlay the introductions that occurred during the colonization of the Caribbean by indigenous people, and here mammals dominate these introductions. The organisms being introduced at this time include things like the hutia, which is a rodent related to the guinea pig, and most of these animals were introduced likely as food sources. There's one yellow line in the reptiles, and that is the green iguana, which was also used as a food source by indigenous people. All of these introductions are coming from the American continents or from other places in the Caribbean. We see a continued increase in introductions during the colonial period and start seeing more taxonomic breadth. Species are being introduced not only for food, but also because now there's this explicit desire to design new ecosystems. And that's exemplified by naturalists like Felix Herminier, who collected fauna from Puerto Rico and West Africa and intentionally introduced them to the island of Guadeloupe in some early acclimatization experiments to see if those animals could survive. Species are also being introduced to make the Caribbean feel more like home to the Europeans who are now occupying it. They brought songbirds, they brought animals that they could hunt as game, and we can then go to the modern era. When we go here, we see that introduced species are now coming from every corner of the world. This contrasts sharply with the indigenous introductions, where introductions were exclusively from Nearctic and Neotropic regions. This time period sees an increase in reptile and amphibian introductions. That's mostly attributable to the exotic pet trade and the nursery industry. And there's also a lot of intra-Caribbean introductions happening due to the shipping trade that operates between islands. So there are a lot of case studies that we can discuss in the paper. And I'm not going to go through those today, but I do want to summarize some of the ramifications of these species introductions on the Caribbean fauna. And a lot of these have relevance to insular systems worldwide. So one of the most critical impacts of species introductions is the extinction of native fauna. A great example of this is the mongoose that I had mentioned earlier, which was introduced to the Caribbean during the colonial period as a pest control agent on sugarcane plantations, but didn't really do that job. Extinction combined with colonization leads to a process called homogenization, meaning that previously unique biogeographic realms become more similar to one another because we lose those unique endemic species and we gain cosmopolitan taxa. Paleontological data helps us understand the timing of homogenization because we have baseline data for what a community looked like before any type of human arrival. Then we can also compare that baseline data to how the community is changing when we start adding these different human groups to the Caribbean and seeing how they're changing the landscape. There is also this exciting and interesting question that develops that maybe is a little bit more hopeful about the restoration of ecosystem services with some of these introduced species. So as I mentioned earlier, the Caribbean lost the majority of its mammalian fauna during the Holocene. Mammals like sloths and hutias and bats, which are still present in the Caribbean, all provide really important ecosystem services like seed dispersal and pollination. The introduction of new mammals as well as birds may serve to restore some of these ecosystem services. But this is something that needs further investigation as to whether these introduced species are producing any net benefit. Finally, we can also think about removing introduced species, a process called eradication. and reintroducing native species to areas where they may have been extirpated, or introducing closely related species to areas where an extinct species once was found. These activities fall within the purview of restoration biology, where the ecosystem is manipulated to recover a previous biological state. Many government and NGOs have been actively working to restore ecosystems to previous biological states. That certainly is something of interest globally and in the Caribbean. Such efforts have helped to stop population declines in some native species, and we have been able to restore some ecosystem services and ecosystem functions through this. But we have to remember that these eradications and reintroductions aren't always feasible because of financial constraints and geopolitical boundaries. So if we have a species that's distributed across the Haitian and Dominican Republican border, and we're able to do some efforts in the Dominican Republic to eradicate a population, we still might get a reintroduction because of adjacent populations in Haiti. Also, these are dynamic systems where many extinctions, extirpations, introductions have taken place over the past 7,000 years. So introducing an extirpated species might lead to some novel interactions with introduced species that may have negative impacts on native biodiversity. So trying to introduce an animal to increase seed dispersal may result in that animal providing seed dispersal services for an introduced species that you do not want to compete with your native species. One major takeaway from the work that we did on this project is that humans have been and will continue to be ecosystem engineers, and we must take this into consideration when thinking about restoration baselines. So pre-European baselines are often thought to represent pristine, non-anthropogenically altered environments, but that's not the case for most of the places we study around the globe, and certainly the Caribbean is just one example of that. So for us to understand what communities looked like before human colonization, we must turn to paleobiology. So in the last part of my talk, I want to focus on what's happening with a lot of species in the ecosystems that I study. A lot of them persist, and sometimes they will diversify when faced with global changes. So I'm going to take this opportunity to highlight some of the research that we're just starting to pick up speed on that's happening in Texas. Because while we have been able to engage with these questions in the Caribbean, some of the paleontological sites that we have access to and data from in Texas provide, they have unique qualities that make it easier to delve in this topic that the Caribbean systems don't have. Much of the work that we've been doing over the past few years since I arrived at UT has centered around a locality that is well known to many of the people in the Jackson School. That locality is Hall's Cave. So Hall's Cave is located on the Edwards Plateau, and it's very well known among North American quaternary paleontology sites. It has a relatively continuous paleontological sequence spanning the past 20,000 years, which is amazing. It's a very fossiliferous site at which the mammalian fauna have already been well described. Much less is known about the reptiles and amphibians, though. So the history of scientific inquiry parallels many previously excavated sites in the Caribbean, which is exciting to me. It's very well dated, and this contrasts with many Caribbean sites where preservation of collagen and biomolecules is poor. So therefore, it's difficult to directly date fossil specimens where you might want to develop last occurrence chronologies for, or just develop age-depth models more broadly. Also, unlike many Caribbean sites, we have paleoclimatic reconstructions based on data from the cave site, but also from other localities on the Edwards Plateau, and that's been very beneficial. Circling back to what I mentioned earlier, Hall's Cave presents an opportunity to evaluate the persistence of reptile communities on continents and start getting at this question of whether extinction rates are higher on islands than on continents. And there's a much larger literature here that I'm not going to delve into today because of time constraints. But we've started chipping away at querying the reptile and amphibian fossil record of Hall's Cave and Central Texas more broadly. And that work is really being spearheaded by one of my graduate students, David Ledesma, who some of you may realize he completed his undergraduate degree in the Jackson School and is now crossed the street in integrated biology with me. He's been studying the reptiles and amphibians of Hall's Cave for his PhD. And to date, much of his work has simply been identifying the fossils, which is a pretty serious task. There are thousands of bones that he's been identifying, often going up to the Vert Paleo lab at Pickle to do this work. And these are just some of the reptile, amphibian, skeletal elements that he's been able to identify in these vials labeled scrap bones, so vials that people just kind of put aside and didn't pay much attention to. He's identified a few taxa at Hall's Cave that haven't been described there previously, which is exciting. And the morphological identifications that David is performing serve as a foundation upon which subsequent ecological and evolutionary analyses can occur that we're extremely excited about. So more specifically, some of his future directions include identifying specimens to the lowest taxonomic level possible using morphological characters and geometric morphometrics, which are both pretty standard techniques. But then also he wants to apply ancient DNA to this system, and preservation at Hall's Cave is so great, unlike in the Caribbean, we're actually able to do some of this work here. And then using that ancient DNA data as well as morphological data from the specimens he's handling, we want to track species diversity, morphological diversity, and genetic diversity through time. We have some species that are extirpated in the system, some that persist, so we can then compare and contrast those responses in this changing climate. We've also started to venture into other taxonomic groups. So we did some really cool work with bats, spearheaded by my master's student, Molly Moros. She conducted a morphological analysis of dentaries for 2 bat species that were present in Central Texas during the Quaternary. So one of those species, Myotis velifer, is still present in Central Texas today. The other species, Aptesicus fuscus, was extirpated about 2,500 years ago during a period that was much warmer than today. So she found that only Myotis velifer exhibited morphological variation across temperature and precipitation regimes. And she actually looked at Hall's Cave and a few other localities in the Edwards Plateau. And the shape variation that she observed occurs in the coronoid process, the condylar process, and the mandibular symphysis. And these parts, if we think about it biomechanically, they all play a pivotal role in bite force. And what this may mean is that changes in these features might relate to changes in the diet and the hardness of insect prey that are available to the bats during these different climate regimes. So this is very much a distillation of just one of the key findings of this work. And there were a lot of other complex patterns that we observed that we are still trying to make sense of, even though we have part of it published or in press to be published. And in some ways at this point, we're left with more questions than answers, but that's exciting because we can now delve into those new questions. That said, one thing that's abundantly clear is that these 2 species are exhibiting different responses to environmental change, and there are different outcomes here. So as I mentioned, one of the species, Aptesicus fuscus, has been extirpated, and the morphological variation exhibited by Myotis velifer may have allowed it to persist in Central Texas, whereas the other one, that was extirpated, maybe it's because of this inability to have morphological variation. It also might just be due to the fact that it responded to this environmental change by leaving Central Texas. So Aptesicus fuscus is actually pretty widespread throughout North America today. And there's just a glaring hole in its range that occurs in Central Texas. And there have been ecological studies of this species, which have indicated that it only really uses caves during cold periods. So as Central Texas became warmer, it may have left the region altogether. Caves are no longer really suitable habitats. And I think it's worth noting that this is an extremely viable and equally resilient response to climate change. Leaving a area and having a range shift isn't necessarily a bad thing. So we're hoping to query these responses further using other techniques, potentially stable isotopes, potentially ancient DNA, all of which can allow us to look at genetic diversity, dietary diversity through time. We can start querying the connectivity of these populations to one another, as well as to other parts of the country, which would be very exciting. And these projects feed into some exciting collaborative work and also interdisciplinary work that we're conducting at UT right now through Paleo Texas, which is a Planet Texas 2050 Grand Challenge project. We're synthesizing paleoecological data with archaeological and paleoclimatic data from Central Texas to identify synergies and gaps for further inquiry, to get at questions like how has environmental change impacted ecosystems and the humans who reside in ecosystems in Central Texas over periods that include abrupt warming. And we're hoping that some of our insights into these past responses of humans and ecosystems may be informative for future responses, because Texas is expected to get warmer and drier in the future. So now just to conclude, paleobiological data provide evidence of non-random quaternary extinctions in a host of taxonomic groups. And what this means for thinking more broadly, trying to contextualize this, ongoing and future extinctions are also likely to be non-random. And paleobiological data might provide insight into the extinction risks of extant taxa based on the life history traits that they have and the pressures that they face. Also, paleobiological data coupled with archaeological data help to disentangle species introductions and species interactions. And from a conservation perspective, this can be really helpful in informing restoration baselines and the feasibility of restoration targets. Paleobiological data allows us to reconstruct phenotypic variation over long timescales in both extinct and extant lineages. And finally, paleobiological approaches to studying the Anthropocene are interdisciplinary and uniquely equipped to address the societal and scientific issues associated with global change. And with that, I would just like to acknowledge my lab members, past and present, collaborators I've had over the years from all over the place, really, and also the Paleo Texas team who I've been working with on more recent work that I'm very excited about.
Fabulous talk, very interesting. But I think we can let it go to now. I think we've stayed on. Thanks everyone for attending our first Afford. Thank you so much, Dr. Kemp, for your fascinating lecture. And I hope to see everyone next week.